So friends, welcome to unit 2 of AGB second professional year. So in this unit, I will be taking lectures on population genetics. So today I will start with the introductory part of population genetics. So in introduction, first of all, we have to know what is population and what are the individuals and what is the meaning of population, and thereby in this way we will learn what is the difference between individual and population. So how individual differs from population, and if we see the comparative study of individual and population, so that we can get a better understanding of individual as well as population, because we know the collection of individual forms population. So population, the individuals are the integral component of population. So these two are inseparable things, but we have to, at the same time, we have to differentiate between the individual and population. We have to differentiate between the characteristic of individual and population. So let us start with the first. We'll see what is population. So what is the meaning of population? So in genetics, population, in statistics also I have defined population in a different way, but in genetics terms we can say genetically a population is defined as a group of sexually interbreeding or potentially interbreeding individual. So these are the group of individual who are either sexually interbreeding or they have potential of sexual interbreeding. So such group of individuals are known as population. If you want to know about the Mendelian population, then it refers to the sexually reproducing and cross-fertilizing group of diploid individuals. So the Mendelian population refers to sexually reproducing diploid individuals that share a common gene pool. So this sharing of common gene pool is very essential. So that means the Mendelian population, individuals in the Mendelian population, they share a common gene pool. So the question comes, what is gene pool? So you have already studied, you have prior information of gene pool because you have studied about the gene pool in your +2 or during the preparation for pre-medical entrance test. So here we will see some minute details of the gene pool. Gene pool means the totality of allele at a particular locus. That means you are having a particular locus, and that locus there will be multiple such kind of locus in a population. The locus will be, so many locuses will be there. That particular locus, suppose a particular locus which is governing a character of flower color. Flower color is governed by a gene or allele which is residing in a particular locus. So suppose there are many plants, so there will be multiple copy of that locus. So gene pool means the totality of alleles at a particular locus. So on that particular locus, how many alleles are residing, or the total alleles which are residing on a particular locus is called as gene pool. Or in other words, you might be aware about this definition. It may be the sum total of genes in the reproductive gamete of a population. So sum total of gene is also known as gene pool. So Mendelian population refers to sexually reproducing and cross-fertilizing group of diploid individuals that share a common gene pool. So this is the difference between population and Mendelian population. Population is the broad term and Mendelian population is the particular characteristic of a population. Now, in population genetic portion, so the first portion in unit 2 of this second professional year AGB course is divided into two parts. One is the animal genetics portion and another is the population genetics portion. Principle of animal genetics, and another portion is the principle of population genetics. So the simple principles of animal genetics, in that portion you will be studying about the uh, basic principles of animal genetics, like Mendelian's different Mendelian's law, exception to Mendelian's law, monohybrid cross, dihybrid cross. That means there you will study the inheritance pattern, or in a study of inheritance of a gene which is governing either one or two or three or few characters in a few individuals. But if that principle we are extending to a population level for a number of traits, then such study is known as population genetics. So in that terminology series, second we want to know about the difference between idealized and real population. What is idealized population and what is real population? So whenever the term ideal comes, that means ideal actually doesn't exist, or it is very rarely existing. So an ideal population is one which produces progeny with all possible relationship between united gametes including selfing. So what it means to say that in ideal population there is possibility of every kind of interbreeding. That means interbreeding between all the relationships. That means distantly related individuals are mating, closely related individuals are mating, even brother sister mating is also there, father daughter mating is there, son mother mating is there, and even selfing is also there. That means individual, both the male and female gametes of same individual is uniting together. So this selfing, it is common in plants and few lower vertebrates, but it is not common in, or it is not possible in case of animals. So in animals, idealized population will refer to a group of individual where progeny are produced due to mating of individual with all possible relationship. So the condition for idealized population is this ideal population is a congregation of subpopulation or line. These ideal population is divided into different lines or different subpopulation. Then secondly, the number of breeding individuals are same for all the lines. So all the breeding individuals are present in that particular all the lines. And third one, there will be no overlapping of generations. So these are the three main criteria of idealized population or condition for idealized population. A population is said to be idealized population if these three conditions are met. Now what about the real population? So real population will have overlapping generation. That means in reality, if you see a population, if you see a population of deer or lion or human or cattle, so suppose you are taking a population of cattle in a farm. So suppose in a farm there are 200 or 500 cattle. You will see every stage of the cattle, every age, the cattle of different ages are there in a farm. They are existing together. That means father is there, grandfather is there, grandmother is there, granddaughter is there, daughter is there. Means at least 3 or 4 generation of animals are kept together. So that means in real population there will be overlapping of generation. There will be in real population there will be migration and selection, etc. So in total you can say that idealized population is one which produces progeny with all possible relationship, and it is having these three conditions. That is, the population is a congregation of subpopulation or lines, and number of breeding individuals are same for all the lines, and there will not be any overlapping of generation. In addition to this, another condition is that there will not be any migration, mutation or selection in case of idealized population, whereas real population states that there will be the individuals in that population will be, or that real population will have overlapping of generation, and there will be migration and selection. Now we will come to the major point of discussion that I have to discuss today is the difference between individual and population. So we are differentiating the characteristic. We are studying the characteristic of individual and population on the basis of few parameters. So first one is the genetic makeup. So individual is characterized by its genotype, and each individual has only one type of genotype, that is either homozygote or heterozygote. So how you will characterize individual? You will characterize individual on the basis of genotype. So whenever you are seeing an individual, if you see genetically, that individual will be a genotype. You have studied the definition of gene, allele, genotype, phenotype, etc. So now what I wanted to tell, that if you see an individual, that individual is actually a genotype. In genetics, if you view it in the from the eye of a genetician, you will see that individual is actually a genotype. Whereas if you see a population is a permanent collection of genes. What you are seeing is population, actually it is a collection of genes, and that is collection of individuals, and individuals are actually genotypes. So the population can be defined or can be described in terms of gene frequency, genotype frequency, etc. So it will have all the alleles at a locus, and allele frequency may be ranging from 0 to 1. So in a population, there will be individuals of different genotype and carrying different alleles, and you can define population in terms of gene frequency and genotype frequency, and that gene and genotype frequency or allele frequency may be ranging from 0 to 1. That means either a particular allele will be there or it will not be there. So if it is there, if suppose only one allele is there, then the allele frequency of that particular allele will be 1. And if suppose a particular allele is absent, then the frequency will be 0. That means gene frequency and allele frequency will be ranging from 0 to 1. Maximum frequency will be 1 and minimum frequency will be 0. So second is the change in genetic makeup. Now you define the individual and population in terms of genetic makeup. Second is the change in genetic makeup. How changes will be there in genetic makeup of an individual. So there will be very rare change in the genetic makeup of an individual. Our genetic makeup of an individual is fixed. It will not change until and unless there will be rare mutations. So mutations are very rare. So individual is having fixed genetic makeup. Whereas if you see the population, actually the genetic composition of population is changed over space and time due to mutation, migration, selection, mating system, and size of population. So these are the factors that causes the change in genetic makeup of a population. So it is actually a congregation of individuals, and this gene frequency and genotype frequency will be changing over space and time, and that is due to mutation, migration, selection, mating system, and size of population, etc. So these are the two characteristics we have seen. Now the next one is the succession of generation. So there is no replacement of an individual itself. And if an individual has taken birth, it will die. And after die, there is no way for getting exact replacement of that particular individual. If individual will come and go, but that particular individual will never be replaced by another individual. So there will not be any succession of generation for individual. But in case of population is getting replaced through generation by the way of birth and death of individual is a continuous process. So succession of generation is common, or it is a characteristic of population but not of individual, because the individual deer, suppose there is a population of deer, so individual deer will die and it will go on forever, but if you talk about a population, the population of deer will be replaced. A few animals will die, another animals will take birth. Then this will be a continuous process. That means succession of generation is there in population. Another characteristic is single versus cluster of measurements. So when we are talking about an individual, the individual is described in terms of single measurement. Single measurement, either we are taking the weight of an animal, height of an animal, or milk yield of an animal. Whereas if you see a population, so how you are defining a population? You are defining a population by taking list of such measurements. And if you see the list of such measurement, you will be confused or you will not be getting an idea. So what you will do, you will put your data to certain statistical analysis or such as measures of central tendency, measures of dispersion. So these kind of measurements will be applied in case of population, but for individual there will be single measurement. So this is the difference for individual and population for fourth characteristic, that is single versus cluster of measurements. So for individual there will be single measurement, whereas for population there will be cluster of measurements. Then fifth one is the lifespan. So an individual is having fixed lifespan. An animal, if it has taken birth, it will die after a certain period of time, and that period is known as lifespan. So lifespan of an individual is fixed. That means an individual is mortal, whereas population is immortal. Population, birth and death is a continuous process in population. So that means a population is practically immortal with no definite date of beginning or end. So when it was started and when it is going to end, no one can say. That is why the lifespan of population is not fixed, it is a continuous. Now the sixth characteristic is the integration and area occupied. So individual can only be present in one place at a time. So a particular individual will be always present at one place at a time, and its physical boundaries are defined. So the physical boundary of an individual is defined. Whereas if you see a population is spread over a limited or wide area with indefinite boundaries, showing geographical gradient and composing local subpopulations. So a population will be divided into various subpopulations, and that may be extending to a large geographical areas. So this is about the population and individual in terms of integration and area occupied. So the last characteristic is the tolerance range. The individual can live or reproduce only within a certain limit of physical factor in its environment, that is called as tolerance range. So tolerance range of an individual will be limited. A particular individual may be comfortable within a particular temperature range. Suppose say from 5 degrees Celsius to 45 degrees Celsius or 48 degrees Celsius. So that range is for individual. Whereas if you see a population, the tolerance limit will be vary to certain extent for individual, which make the tolerance for a population than any individual. So if you are talking about population, in that population there will be many individuals who are very well adapted to lower temperature, and there are few individuals or many individuals which will be comfortable at higher temperature. So tolerance range for population will be larger or higher. The tolerance limit for population will be varying. It will not be fixed for as in case of individual. So these are the 7 characteristics on which you can differentiate or you can study the difference between individual and population. So I think the difference between individual and population is clear, and if it will be asked in examination, you will be able to write it in the form of a short answer. Okay. The next one will coming, actually this population genetics, actual population genetics I will be discussing from next lecture onward. But just for introduction, what is population genetics? So population genetics is concerned with the statistical consequences of Mendelism in a group of families or individuals. It studies the hereditary phenomena on a population level. So as I have already told you that what is population genetics, it is an extension of Mendelian genetics to a population or group of families or group of individuals. That means it is to a population. If you see here in this diagram, you can see, we know the Darwin has given the evolutionary biology and Mendel has given the lays the foundation of molecular biology. The place where these two join together, that is the beginning of population genetics. That is the beginning of population genetics. That means evolutionary molecular evolution in terms of inheritance. Evolution in terms of inheritance. So now few basic terms that you have already studied when you were studying about the Mendelian genetics. So what is gene frequency? So gene frequency is the proportion of alleles of a gene in a population. So it is a proportion of allele of a gene. Suppose a particular allele is having suppose we are having three individual, 1, 2 and 3. 1, 2 and 3. One is having capital A genotype, second is having capital A small a, and third is having small a. So what will be the frequency of small a gene? So small a gene, in this case, the frequency is 2 divided by total number of alleles. Total number of alleles are 2 plus 2, 4 plus 2, 6. So 2 by 6, and if you simplify it, it is 1 by 3. So this is the gene frequency, or it is also called as allele frequency. Is the proportion of an allele of a gene in a population. So this is how we are finding the gene frequency, and similarly we can find the genotype frequency also. So genotype frequency refers to population of a particular genotype in a given population. In the same way we can find the genotype frequency. Another term is random mating. That is also we have seen when I was discussing about the Mendelian genetics or Mendel's law. Random mating is also known as panmixia. So in case of bisexual organism, it means that any one individual of one sex is equally likely to mate with any individual of the opposite sex. So what random mating means? It means that any one individual of one sex is having equal chance to mate with any individual of the opposite sex. So a population that is mating at random is known as panmictic population. Then, so this is the end of today's discussion. Actually today we have just introduced the population genetics. Actual population genetics I will be discussing in the next class. So thank you very much for your kind attention. Thanks.